I'm Brian McCourt, president of the McCourt Foundation, and we'd like to welcome you all here today for the first ever virtual McCourt Foundation educational update. We've got a fantastic lineup of physicians that we'll be presenting today. Physicians that are not only top in the country, but top in the world in their respective disciplines. We're excited about having you all here and hosting you, and would also like to thank all of our sponsors that are making this possible. But I'd like to especially thank our presenting sponsors, Bristol Myers Squibb and UBS Financial, as well as our silver sponsor, Sanofi Genzyme. But I would like to thank all of you for being a part of this day. This has always been one of our favorite and most impactful programs, and it's all because of you. So enjoy the program. I'd like to now introduce Stephen Sypek with UBS Financial.
Thank you, Brian, and thank you for joining us today, and welcome to the McCourt Foundation Educational Symposium. My name is Steve Sypek, a financial advisor with UBS Financial Services here in Rockland, Massachusetts. I've been friends with the McCourt family for many years and personally involved with the foundation for over 20 years. I too have been impacted by neurological disease, and that my mother passed away from Parkinson's 5 years ago this year. I am excited for today that you will be hearing from the top doctors and researchers from Massachusetts General Hospital, Brigham and Women's Hospital, and Cedars-Sinai Medical Center on their progress and developments in the areas of multiple sclerosis, Alzheimer's, and ALS research. We all know the challenge that individuals and their families face in dealing with neurological diseases. And that is why I believe the McCourt Foundation focus on making things better by supporting research, providing education, and developing strategic health-focused partnerships will one day lead to a cure for many of these diseases. As a financial advisor, I know the challenges that these patients and their families face in planning for the unexpected and in dealing with the emotional, physical, and financial challenges. We believe that the financial services industry can help families plan for those unexpected circumstances, and also help support the critical research and awareness in the areas of neurological diseases. And that is why UBS is excited and proud to support the McCourt Foundation in their great work and their mission by sponsoring today's educational update. Thank you for joining us. I hope you find these sessions helpful and that they will continue to inspire and empower you and your loved ones in making things better each and every day. Enjoy the upcoming update.
Hi, I'm James Barry. I'm standing in for Merit Zakovich, who sent her regrets and isn't able to be with us today. I'd like to welcome everybody back from the break. I know this has been an exciting day so far. Thank you all for joining us this afternoon. We're looking forward to another exciting session. We'll be talking about ALS and ALS research. In particular, I'd like to thank, before we get started, the McCourt Foundation. We're grateful to have the opportunity to come together as a community today to talk as a whole group. The remaining time today will flow as follows. I'm here with two really incredible colleagues of mine from Mass General Hospital, Drs. Ghazaleh Sadri-Vakili and Suma Babu. Each of us will be giving a brief presentation highlighting exciting updates about ALS and ALS research, many of which have been made possible through the support of philanthropic partners such as the McCourt Foundation. Our presentations will be followed by a question and answer session. Now, I'm going to speak first, and again, my name is James Berry. I'm the Winthrop Family Scholar in ALS Sciences and Chief of the Division of ALS and Motor Neuron Diseases at Mass General Hospital. Today, I'll talk about the Healy ALS Platform Trial and Expanded Access Program, and how we can reimagine and accelerate the discovery of new therapies for people with ALS. Now, before I dive into the platform trial, just a few words about amyotrophic lateral sclerosis. This is a neurodegenerative disease. It's primarily characterized by its effect on upper and lower motor neurons, residing in the brain, lower motor neurons in the spinal cord. Classically, this is described as a painless progressive weakness, although that belies the truth of the illness in more progressed states at some point. It's characterized by an asymmetric onset and a regional spread. In other words, it often starts in one limb and can spread through that limb into an adjacent limb before spreading to other parts of the body. It may also start in or around the mouth in what we refer to as the bulbar region. The disease spares eye movements and bowel and bladder function until very late in the disease if they are affected at all, but they can be affected. The average survival after symptom onset is 3 to 5 years, but there's a very wide variability, and there are some people who survive more than a decade and even long beyond that. The average time from symptom onset to diagnosis is 12 to 14 months, which is a difficult reality of the diagnostic process and something that Dr. Babu will talk about later. A longer delay actually correlates with slower progression later in the disease. About 10% of ALS is what we call familial or caused by a mutation in a single gene, but we found these causative mutations in over 30 to 50 genes. 90% of ALS is what we call sporadic. In other words, we don't have a genetic cause of it, a single genetic cause of it anyway. And the last and really important point is that there is a blossoming research and clinical trial landscape in ALS. Now, we think of ALS as an uncommon disease. Some people call it a rare disease, but the reality is that it affects 1 in 300 men and 1 in 400 women throughout their life. And that incidence is increasing globally, in large part because it's more common in older people. We see about 5,000 new diagnoses of ALS every year in the United States, and there are about 20 to even some estimates are 30,000 people living with ALS in the US. The average age of onset is 55, and there are 4 FDA-approved treatments. Now, it's important, I think, to look at the timing of those FDA-approved treatments because we are making headway, and we're making headway at a faster and faster clip. So in 1995, riluzole was approved to slow ALS progression, the first disease-modifying therapy. In 2010, Nuedexta was approved to treat pseudobulbar affect for ALS, sort of an abnormal laughing or crying that can come with the disease, and was very difficult to treat prior to that. In 2017, edaravone was approved to slow ALS progression. In 2018, liquid riluzole was approved. And in 2020, we saw very exciting positive results from a trial of a drug still referred to by the name AMX0035. And Dr. Babu will talk, I think, a little bit about that later. Now, ALS also has a robust therapeutic pipeline and pressure to move fast. The NEALS Consortium is a group of research sites of over 100 sites throughout the US, and it connects us and binds us together in a mission to find cures for ALS. There are over 160 companies that are working in the ALS space, each with at least one drug, and thousands of investigators worldwide working to find new targets in ALS. At the same time, people with ALS say, like Sandy did, I lost the privilege of working on the human time clock when I was diagnosed. The ALS time clock is a lot faster, and we appreciate that and work with that in the front of our minds. The best way to make progress in this situation is to test therapies efficiently, quickly, to find the ones that work. And to that end, we've designed the Healy ALS Platform Trial to really bring together an entire community to rethink the ALS trial landscape so that we can do this faster. Now, when we had this kind of concept, and really it was Dr. Sokovich who really set the stage for this platform trial and has led it. And really the idea was to bring together investigators, industry, FDA as regulators, clinical sites, and put at the center of all of that people with ALS so that we could move rapidly from the concept of this to launch. And we did that much faster than there are platform trials in a few other diseases. Those took years and years to design and launch. We did that in about 12 to 18 months. To explain what a platform trial is, just briefly, platform trials really revolutionize how we construct and carry out trials. So a traditional trial works sort of like if we decided that we would play a game of soccer, and our first step was to sit down and design and build a stadium to play in. That means we'd need to have contracts, and we'd need to put together what we call the protocol or the plan for doing the trial. We need to reach out to all of the sites that may be a part of this. We need to design our schedule of activities and figure out how we're going to monitor safety. All of that happens freshly. And then we run the trial, one intervention for one disease. And at the end of that, we take that entire constructed stadium down. And if we want to play another game of soccer, that is, if we want to do another trial, we start from scratch. There are inherently a lot of inefficiencies there. And it turns out a lot of our trials are really very similar once we get them designed. And that brings forward the opportunity to do a platform trial. In this trial, we build our stadium once. We design a trial that is robust, that will allow us to test medications, test a number of medications, that has very robust safety profiling of drugs. And then we can run not just one drug through that, but 2, 3, 4, 5. And when one finishes, we can add another one. So the way that that works in the Healy ALS platform trial is that the trial is governed by what we call a master protocol. This is the instruction book or the recipe for the trial. It's a common protocol for multiple therapies, and that's what sets it apart. The protocol defines really the global schedules for the trial. When will people come into the clinic? What will we draw their blood? How will we monitor their safety? How will we tell whether a drug is working? What sort of outcome measures will we have? And within this master protocol, we put in what we call treatment regimens. And these are the individual therapies being tested in the trial. The regimens are added to the trial so that there are always numerous therapies being tested at the same time. When the Healy platform trial began, it began with 3 regimens, different drugs, all being tested at the same time. And we've since added more, and we'll continue to do that. There are several benefits to a platform trial, and the Healy platform trial in particular. Our estimates are that we can cut costs by at least 1/3 for each regimen, and we can cut time in half to find an effective therapy. Perhaps one of the most exciting options or benefits of the platform trial is that we can run this trial and we can offer it with less placebo and more options to participants. So when a participant decides to enroll in the platform trial, he or she would be randomized to any of the ongoing regimens. Here I'm showing the first 3 regimens, A, B, and C, but later they might be F, G, and H. They'll be randomized to a regimen. They will know which regimen they're randomized to. And then within that regimen, they'll be randomized to placebo or active study drug. Now, normally, there's a 50-50 chance of someone getting placebo, which is an inactive matching sugar pill, people often call it, or the active study drug. But in the platform trial, because of the way we've designed it, we can actually increase the chances of getting active study drug so that there are 3 people getting active study drug for every 1 getting placebo. And in the end, because we need to have a balanced number of people on active and placebo, we then share the placebo, pool those placebos so that we have 1 placebo group from each of those regimens or 3 times the placebo, and we can compare that to each regimen. This is an efficiency that really sets the platform trial apart from any other design. We can also leverage the platform trial to learn more about ALS and to build better tools to hasten future ALS trials or future regimens within the platform trial. So we can sequence DNA and learn new insights about ALS and about pharmacogenomics during the trial. We can look at well-healed biomarkers like neurofilaments, but we can also explore new biomarkers in blood, urine, and spinal fluid. And finally, we're using speech and digital monitoring to try to find less invasive, faster, more objective, and easier to do outcome measures to make the trial overall simpler for participants. Now, the trial has 54 sites. In general, in the past, each site would have an ethics board overseeing the study, each ethics board with its own set of questions. And that slowed down startup of sites dramatically. We now have one central ethics board that we often call the IRB. And that ethics board asks its questions, makes sure that the trial is run ethically and safely, and that flows down to all of the 54 sites, bringing a huge amount of efficiency. Now, I said that we started with 3 regimens. We've since added a 4th regimen. Those regimens are zilucoplan, verdiperstat, CNM-Au8, and pridopidine. Again, each of these are randomized 3-to-1. And at the end of a 6-month randomized portion of this trial, there is an open label extension for all of these. And that open label extension ensures that people who've been in the trial, whether they were randomized to placebo or active treatment, all have the option of receiving active study drug at the conclusion of their randomized portion. Now, the 5th regimen has already been added and is in its design phase. That's Trilahos by Selos. And we're in discussions with at least 2 more companies for 2021, if not more. Each of the treatments that goes into this trial is selected by a committee of ALS scientists and experts who really vet it to make sure that the science behind it is exciting. Now, all 54 sites have ethics board approval. It's a centralized approval process. 44 of those sites are actually enrolling participants now. We've enrolled 323 participants, and 217 are already receiving study drug and placebo. The remaining ones are in their screening period and will soon start the drug or placebo. There are 30 plus more sites that are interested in joining the trial. And one of the additional benefits of this platform trial is that the geographic spread is much greater than most trials that go on at just a handful of sites, limiting the access to people with ALS to these trials. Now, these first 3 regimens are 50% enrolled, and the trial is enrolling faster than any previous or current trial in ALS. It is really remarkable. This is a list of the 44 active sites that are enrolling as of a couple of weeks ago. And again, there's a spread across the country of each of these sites. Now, we know that this can be dizzying, so the platform trial also has a patient navigator, actually 2, Catherine Small and Allison Boulat. And they can be reached at 833-HALT-ALS by anybody across the country who has questions about how they might get involved in this trial, what's their nearest site. They can also be reached at this email address. All of this is available on the Healy ALS Platform Trial website as well. Now, we have a companion effort, which is the MGH ALS link, which is also available on our website. You can find the Healy ALS website by simply Googling Healy ALS or MGH Healy ALS. And the MGH ALS link is really an email list that you sign up for where we send out newsletters and opportunities to participate in ALS research and new information about ongoing or completed trials. I want to turn my attention for the last couple of slides here just to talk about expanded access protocols and how they're giving us new ways to include people with ALS in research who really in previous years were excluded. These expanded access protocols are FDA regulated, and they're designed to provide people with serious illness, including ALS, access to experimental therapies. And we're able to provide expanded safety data about experimental therapies by using data that we collect from expanded access programs. But these are aimed at people who are not eligible for clinical trials, perhaps because they've had disease too long, perhaps because it is more advanced, or they just don't have the characteristics of a person who would fit into an ongoing trial. And for that reason, they're not able to participate in this really hopeful and I think important endeavor to move the field forward. But expanded access programs are meant to fill that void. They're sort of situated at the nexus of clinical care and research. They're not trials, and yet they're not typical clinical care because they are, we hope, a source of hope and a source of excitement, but at the same time, these are experimental therapies. Now, the last, I think, important part is that these are often a partnership between industry partners and academic partners and patients. They are usually supported, at least in part, by philanthropy. So, another example of really critically how philanthropy supports what we do. The Healy Center has really had a number of expanded access programs with a handful of participants in each. But over the last couple of years, we've been able to grow the total number of enrolled participants nearly up to 100 participants. This is really what we kind of dreamed of when we began these programs, and you can see they got off to a slow start and have been going much faster recently. So this is really an opportunity for people who otherwise would not have access to experimental therapies. A new program is the Healy ALS Platform Trial Expanded Access Program. And this is a collaboration with Biohaven and funded in part by IMALS. It's an expanded access program of verdiperstat, which is the platform trial regimen that's sponsored by Biohaven. And in this EAP, all participants will receive the study drug. The protocol is similar to the platform trial, but simplified so that we don't have to do as many visits, although we have to certainly monitor the safety in the same way. There are 3 platform EAP centers, Northwestern, Duke, and MGH, and this really sets it apart from the expanded access programs that I was talking about earlier, which were all single center. The plan is to have 30 participants, and it will be open to people ineligible for the main platform trial. It is in startup currently, and we look forward to this getting started. So just to summarize, we really hasten progress through scientific discovery, partnership, efficiency, and inclusivity. Basic researchers are creating breakthroughs in ALS science. These breakthroughs are translating to novel therapeutics. We're developing new ways to test these therapies more efficiently than ever, like the platform trial, and in some ways, the expanded access programs. We're working with the FDA, with industry partners, with ALS clinicians, and with people with ALS and their caregivers to ensure that new trials are better designed than ever, simpler, more available. Expanded access brings experimental therapies to a broader range of people with ALS than ever before. And with all of these new tools and everything that we're learning about ALS, the future is bright. And you'll hear some more about why the future is so bright from our next speaker, Gaz Sadri-Vakili. So I'm going to pause here, and Gaz will begin sharing her screen. While she does that, I just want to introduce her briefly. Gaz is an assistant professor of neurology at Harvard Medical School and at Mass General. She directs the Neuroepigenetics Laboratory at the Mass General Institute for Neurodegenerative Diseases. Thus far, Dr. Sadri-Vakili's laboratory has identified disease-specific alterations in multiple cellular pathways using human samples, as well as cellular and animal models of ALS, Huntington's disease, and X-linked dystonia Parkinsonism. In ALS, her laboratory is assessing the pathogenic role of neuroinflammation, mitochondrial dysfunction, and the epigenome. She works very closely with clinical research colleagues from the Healy Center at ALS to translate her findings in fundamental laboratory research into meaningful breakthroughs for people with ALS. Today, she's going to talk about ALS, the latest research and preclinical updates.
Thank you. I'm delighted to be here. Good afternoon, everyone. My name is Ghazaleh, as James just introduced me, and I'm delighted to tell you about some of the preclinical updates in ALS. If I can get this, there we go. Sorry. So here's an overview of what I'll be discussing today. I will talk about some pathogenic mechanisms, mainly focusing on protein aggregation, trafficking, and a cellular process known as autophagy, giving you 2 different examples on that, and then finishing up by telling you a story about inflammation in ALS that we completed last year. This is a pretty busy slide, but the point I'm trying to make here is that many different things can go wrong in ALS, and you can see them highlighted here with these exclamation yield signs. But what happens is that it's not just the neurons here with the cell body, with the axon. It's not just the neurons that go through these processes or have deficits in their cellular processes. It's also other cells of the CNS, such as the oligodendrocytes, astrocytes, and as well as the microglia, that can also be involved with causing pathogenesis in ALS. And because this is too complicated to look at this way, I decided to summarize these pathogenic mechanisms in the form of this colorful image that you can see here. Now, there are too many pathogenic mechanisms for me to discuss today, so I'm just going to be focusing on the ones that are highlighted here. And the point that I like to make is that a lot of these mechanisms are actually linked. So when we're studying one, it could also be involved in other processes within the cell. I'll start by talking about protein aggregation, which is a hallmark of neurodegenerative disease. Here on the left, you can see that protein aggregates are found in a number of neurodegenerative diseases, including Huntington's, Alzheimer's, Parkinson's, as well as prion disease. And here you can see two examples of protein aggregates being formed in ALS. On the right, I'm showing you the different genes that are known to be linked to cause ALS. SOD1, TDP43, FUS, and C9orf. Mutations in all of these genes or proteins will lead to different forms of aggregates within neurons. So the point I'm trying to make is that protein aggregation occurs across neurodegeneration, but also in ALS. Now, why would this happen? One of the reasons where you can have disrupted protein synthesis, or excuse me, if you can, that you can have the increases in protein aggregation, could be due to a loss of autophagy, which is a cellular trash removal system. So this is one of two different ways that we can get rid of trash within the cell. And it's really very simple. The cell will form a membrane around proteins or DNA or RNA that needs to get degraded. And you can see that happening here, forming what's known as an autophagosome that will then fuse with another organelle called the lysosome within the cell. The lysosome then has enzymes in it and it's more acidic, which will then lead to the degradation of the unwanted proteins. So you can imagine that if you lose your trash service, then you're going to have this pileup of trash. And this is sort of what happens and how proteins could end up, one way in which proteins can end up aggregating in the cells. And what we know is that autophagy is becoming a really hot target in neurodegeneration, as well as in ALS in particular. We know that maybe if we could increase autophagy, then perhaps we could degrade some of these protein aggregates and clear them. But also we're learning that the lysosome itself doesn't fuse correctly to the autophagosome, so there's issues with that. Also, there's problems with the pH within the lysosome. So there are many different areas in which we can target this pathway. And there are a lot of wonderful labs working on these processes. So a new target, I would say that I'd like you to know about is how we can improve autophagy in ALS. And that could be one way to go about this. Another thing I like to talk about is trafficking within a cell. So just like you have this many lane highway going in two different directions, if you can imagine, here is a very simplified schematic of a neuron with the cell body here, the nucleus, and the synaptic terminal on the other end with the axon in the center. There are things known as microtubules that are within the axons, which I like to think about as the highways within the cell, where you can have, where you can transport proteins and different organelles within the cell from, let's say, the cell body all the way to the axon terminal, or in reverse, just like this two-way highway. So this trafficking also occurs between the nucleus as well as the cell body. You can have trafficking of RNA and different proteins within the nucleus and the cell body. So you can imagine that if you have disruptions in any of these areas, you can have some problems. So let's focus on the nucleus first. It's been described now over the past 3 years that there are issues with the nucleocytoplasmic transport in ALS. And one protein that's really affected by this is a protein known as TDP43, which is a nuclear protein involved in transcription and RNA processing. Importantly, mutations in TDP43 have been shown to cause ALS. And TDP43 aggregates are found in 90% of ALS cases, including sporadic cases. So in ALS, we know that TDP43 is mislocalized to the cytoplasm. And if you remember, I said that this is supposed to be a nuclear protein. So a number of our colleagues set out to determine what does loss of nuclear TDP43 lead to. And what they found is when you have a loss of TDP43 that forms these cytoplasmic aggregates, what happens is it directly affects the expression of another protein known as stamin-2, which is a microtubule binding protein. So you could imagine stamin-2 being one of the cars that can take the cargo down your axons into the synaptic terminal. And stamin-2 not only binds to the microtubules, but it's also really important for axonal regeneration and growth. And what our colleagues demonstrated is that in ALS, when you have this loss of nuclear TDP43, you can see it here in red outside of the nucleus. What happens is it leads to this degeneration of axons. And that's exactly what happens in ALS at the neuromuscular junction. We have denervation. So stamin-2 could play a role in this. And what our colleagues are hoping to do is to reverse this and make it more like a normal situation where you have nuclear TDP43. So stamin-2 is a new target now, and our colleagues are working hard to replace it or replenish it and perhaps help in regenerating the neurons and helping with the denervation that happens at the neuromuscular junction. Okay, what else can go wrong in terms of trafficking and bringing together with protein aggregation? You can imagine if you have a pileup on your highway or you have a bunch of proteins aggregating on your microtubules or in your axons, then perhaps you're not getting the cargo where it needs to go, whether it's the synaptic terminal or even in the cell body. So now new studies from my lab are focusing on another protein that's a microtubule binding protein, and it's known as tau. Now, you may have heard about tau if you were listening to the earlier talks on Alzheimer's disease. Tau stabilizes axonal microtubules, is important for trafficking, and it's been implicated in Alzheimer's disease. Not only that, tau has been shown to aggregate in a number of neurodegenerative diseases, and those neurodegenerative diseases are known as tauopathies. So I'm not going to tell you that ALS is a tauopathy. However, we have begun to notice that tau is also aggregated and hyperphosphorylated in ALS, similar to what's known in other diseases. So what we found is similar to Alzheimer's disease, tau is shifting from the cytosol to the synapses. So it's shifting from this area of the neurons to the outer areas. And when that happens in Alzheimer's, we know that this hyperphosphorylated mislocalized tau could be toxic for the neurons. And it could be toxic because it can increase synaptic loss, but it also has been shown to interact with mitochondria. And I'm going to tell you more about that. So we hypothesized based on these preliminary findings and also what's known in Alzheimer's disease that alterations in tau could play a role in mitochondrial dysfunction in ALS. And perhaps if we could target tau, we could reverse these abnormalities. As you know, mitochondria are the powerhouses of the cells. They produce energy, but when they're not working properly, as they don't in different neurodegenerative diseases, there's a decrease in ATP or energy production, there's a decrease in calcium buffering, and also mitochondrial dynamics, their shapes themselves are altered. More importantly, they non-functioning or dysfunctioning mitochondria can increase there's an increase in the reactive oxygen species or ROS, which could end up being really toxic for the cells. So talented postdoc Tiziana Petrosiello in my laboratory started to assess mitochondria and the role of tau in altering mitochondria in ALS. And using electron microscopy, she was able to show that in postmortem brains from ALS patients, there is a decrease in the number of mitochondria. But not only that the mitochondria that were there were actually smaller. So there's an increase in the number of smaller mitochondria. suggesting that mitochondria are smaller and fragmented and perhaps less energetically favorable in ALS, as we know. So we wanted to know what is the mechanism that causes this fragmentation in ALS. And I was just describing to you that mitochondria are very dynamic. They can fuse together and form these larger networks, which are more energetically favorable, or they can go through this process called fission, where they're fragmented and form these smaller mitochondria that could then be degraded more easily. And this can occur through the activity of a protein known as DRP1. What we found when we measured DRP1 levels in postmortem brains from ALS patients is that there was a significant increase in DRP1 levels that went hand in hand with those smaller mitochondria that I was showing earlier. Because of time, I don't have, I can't show you all of this data, but I want to leave you with this, that we found that alterations in tau and DRP1 are common mechanisms across ALS. We've looked at them across a large postmortem sample size, whether they're male or female patients, or if they had TDP-43 aggregates or not. No matter what type of ALS it was, bulbar onset, limb onset, we found these differences in tau and DRP1. So then we wanted to know if the synaptoneurosomes that are enriched in tau and DRP1 could alter mitochondrial dynamics, and we set up an in vitro assay. So we're using cell lines here and treating the cell lines with synaptoneurosomes derived from ALS patient brain or control brains that were from healthy individuals. And we're using here a recombinant tau as a positive control. And when we treat cells with these different conditions, we find that the ALS synaptoneurosomes and the tau alone can cause shattering of the mitochondria. So here you can see that there is a significant increase in the number of smaller mitochondria and that there's a decrease in the number of larger mitochondria. Not only that, we find that there is a decrease in the number of large networks, mitochondrial networks, and that the number of the mean branch length is also decreased following treatment with ALS synaptoneurosomes or tau, suggesting that, again, tau and DRP1 may be playing a role in this. So then we wanted to know if we target tau, if we degrade it, can we mitigate these changes in mitochondrial dynamics? Here, we're working with our colleagues, Dr. Haggerty and Dr. Silva at Mass General Hospital, that are using this new technology called proteolysis targeting chimeras or PROTACs. This is a way where you can degrade your protein of interest. And I think over the next few years, we're going to be hearing more and more about this novel technology. And they have developed a novel degrader where I'm just going to call it QC. You can see it here just to be brief, but QC is able to degrade tau and phosphorylated tau levels. And so we're using that here to treat our cells and show you that we can significantly decrease tau levels following treatment with QC. So we wanted to know, if we degrade tau, how does that affect the mitochondria? And as you can see here with these solid bars, the solid bars are now the ones that got treatment with QC. We're actually able to mitigate the effects of tau or ALS-derived synaptoneurosomes. So by degrading tau, we're able to decrease the number of smaller mitochondria and also increase the number of larger mitochondria. And the same thing we're seeing with the networks, we're able to reverse the effects of the ALS synaptoneurosomes and tau by removing tau and restoring the mitochondrial networks. Now, does that have any functional effects or not? The answer is yes. So I showed you, or I told you earlier, that when mitochondria are dysfunctional, there is an increase in reactive oxygen species, or ROS. And what we're able to do is, when we degrade tau, then we can restore or decrease the amount of reactive oxygen species that were increased in response to the treatments. So in summary, we think that phosphorylated tau and DRP1 interact together in ALS, causing the formation of smaller mitochondria and increasing oxidative stress. And if that we target tau, we're able to increase the size of the mitochondria or get rid of the smaller mitochondria, and also decrease oxidative stress and reactive oxygen species. And in the last minute, I just want to touch upon inflammation or neuroinflammation, which is also one of the pathogenic mechanisms that we talk about in ALS. We know that inflammation occurs and it's important for disease progression in ALS. A number of different inflammatory markers are altered in ALS. And here on the right-hand side, I'm showing you a brain from an ALS patient versus a control. This is a PET scan that's specific for a marker of inflammation. You can see the more red and yellow you see, there's more binding of that marker to the brain. You can see that happening in a patient living with ALS, that there's more inflammation in their brain compared to a healthy control. So we have different ways of monitoring this and measuring it. So the thought is, if we could mitigate this, if we could decrease inflammation, perhaps we could help with neuronal survival. This is, again, a busy schematic, but what I like to show you is those microglia that I talked about in the beginning, or the glial cells, they can become reactive and cause more inflammation. And we know in ALS that there is an imbalance between the homeostatic glial cells that are supposed to be there supporting the neurons, and the ones that we call reactive, which end up releasing a lot of pro-inflammatory cytokines and chemokines that could cause neuronal death and injury. So if we could balance this out to decrease these reactive glia and make them more homeostatic, then perhaps we can balance it. We can balance the immune response. And one compound that's known to do this is called cromolyn sodium, that we did a whole preclinical trial in our transgenic SOD1 mice with this drug. And what we showed is that treatments with cromolyn could delay disease onset in the SOD1 mice, that it was neuroprotective and it spared the spinal motor neurons, and it also maintained innervation at the neuromuscular junction. Importantly, cromolyn was able to reduce inflammatory markers and mast cell activation, and mast cells are actually a direct target of cromolyn. And this was really exciting because the company that had provided the compound for us, AZ Therapies, started a clinical trial using cromolyn this past September. They dosed their first patient in a phase 2 clinical trial. So this is just an example of what we do in the lab and how we can translate it into actual clinical trials for the patients. And I feel really lucky as a basic scientist to work with Dr. Berry and Dr. Babu at the Healy Center, so that we could translate some of our basic science findings to the clinic. And I will stop there and pass it back to Dr. Berry. Sorry, before I do that, I'd like to thank everyone in my group who does the work, and also my wonderful collaborators at Mass General Hospital, our sources of funding and other collaborators, and of course, patients and their families. And I'll stop there. Thank you.
Thanks, Scott. Really wonderful dive into some of the biology, and hopefully we'll get a chance to talk a little more about some of that in the question and answer session. I want to introduce Suma Babu, who is an assistant professor of neurology at Harvard Medical School and Mass General. Dr. Babu is an active clinician in the Multidisciplinary Als Clinic at the Healy Center for ALS. She has a special interest in developing disease-modifying treatments and in using neuroimaging, including positron emission tomography and MRI scans, to characterize neuroinflammation and other markers of ALS disease mechanisms in people who are experiencing the disease, where our research is really most relevant. Dr. Babu leads several phase 1, 2, and 3 clinical trials evaluating novel therapeutics for sporadic and familial ALS, in addition to her work in neuroimaging. And today she's going to talk about new hopes for ALS, a new era of clinical trials, and novel approaches for clinical care.
Thank you, Dr. Berry. Good afternoon, everyone. I'm excited to be here for this virtual event today. I'd like to give a bird's eye view of some of the exciting developments that's happening in the field of clinical trials, as well as in the field of clinical trial readouts or biomarker development, and also to include some new initiatives for expediting the diagnosis of ALS for patients as well. So this is the agenda for today. As Dr. Berry mentioned earlier, drug development for ALS is rapidly accelerating. And today in the drug development space, there are more than 160 companies developing drugs for ALS. In addition to many phase 1, phase 2, and phase 3 trials that are ongoing, more recently, we learned about 2 successful clinical trials that were published just within the past year. One of them is a gene therapy approach using a drug called tofersen for people with SOD1 mutation ALS. And the second, as you heard earlier from Dr. Berry as well, a drug called AMX0035 for people with ALS. Tofersen is an antisense oligonucleotide. In short, we call it as ASO for people who carry a mutation caused by SOD1 gene. This drug is being developed by Biogen in collaboration with Ionis and several academic institutions, including MGH. In their phase 1 trial that was published just within the past year in the New England Journal of Medicine, tofersen was shown to be safe, and there were some early signals of slowing of disease progression in people who received tofersen at the highest dosage and who carried the SOD1 mutation ALS. So what is tofersen and how does it work? In SOD1 ALS, there is excessive production of a toxic protein, the SOD1 protein, as you heard from Dr. Sajri Vakili earlier. And this clogs up the system within the motor neurons. It is tofersen is a drug that binds to this toxic, faulty RNA that could produce this excessive amount of the toxic SOD1 protein. It blocks it from producing the excessive amount of the SOD1 protein and hence rescues the motor neuron cell. In the trial, the phase 1 trial, people who received the highest dosage at 100 milligrams, compared to people who received placebo, there was a lowering of SOD1 protein to the extent of 33% as measured in their cerebrospinal fluid, compared to people who received placebo who had only a 3% reduction in the toxic SOD1 protein levels. That's very exciting, and what it translates to from preclinical studies is that 33% reduction is clinically meaningful and relevant. What we also learned from this trial is that there were some early signals of slowing of disease progression as measured by ALS-FRS-R or vital capacity changes. You have to keep in mind that this was a phase 1 trial primarily looking at safety and tolerability. And even in the small group, as using as few as 10 people who received the highest dosage, there were group differences between people who received the actual drug versus placebo in terms of slowing of ALS-FRS-R decline, as well as slowing of the vital capacity decline. And when we looked at smaller subgroups of people who had a rapidly progressive disease, the group difference became more exaggerated for both ALS-FRS-R as well as for vital capacity changes. This is very exciting for the field of genetic forms of ALS. Currently, tofersen is being tested in a phase 3 trial, and this trial is ongoing, and we're awaiting the completion of this trial, which probably would happen later this year. So what's next for genetic ALS? Just within the past decade, there have been more than 10 new genes that are discovered for ALS. One could also look at this as 10 new targets that we could target using the antisense oligonucleotide or other mechanisms of gene therapies to help people who have these genetic forms of ALS. Following the footsteps of tofersen, there are additional ASO clinical trials that are currently in trial, actively recruiting, or in the pipeline. The most common gene causing familial ALS in the US, which is a C9 ALS, or C9orf72, in short, we call this a C9 ALS. This ASO program is in phase 1 currently, and Ataxin-2 is another ASO program that has just recently started. And later this year, we're also hoping to see FUS ALS ASO program to start in its phase 1 trial as well. So overall, the landscape for gene therapies and genetic testing is really rapidly expanding. And keeping this in mind, I would encourage all patients on this call to please discuss with your neurologist if genetic testing or gene therapy approaches might be the right approach for you. Moving on to the second positive trial that was published in the past year, also in the New England Journal of Medicine, was the AMX0035. And this is a phase 2 trial. It's a combination of two drugs, phenylbutyrate and Terso or TUDCA. AMX0035 in preclinical studies showed that it can rescue motor neurons based off of the individual component drugs that can work on two different cell organelles, the mitochondria as well as the endoplasmic reticulum. As you heard from Dr. Sajid Vakili earlier, targeting the powerhouse as well as the trash removal system within the cells. And what we learned from this study is that AMX0035 is safe, and it showed that a slower functional decline in people who received the actual drug compared to placebo over a period of 24 weeks. This trial was led by MGH. Dr. Sabrina for this study was of ALS community. The drug was produced from Amylyx Pharmaceuticals at many different sites. The trial was conducted at many different sites within the US. What we learned from this trial in terms of the primary endpoint is that the ALS-FRSR decline over a period of time was slower in people who received the drug. That's the red bar you can see on the slide at the top, compared to people who received the placebo, which is the blue bar who had a greater progression over the trial duration. And this difference between the drug and the placebo groups was statistically significant at the end of the trial. Further, what we learned is in the open label extension, there was a survival benefit in addition to people who received the AMX0035 who were randomized to this drug in the original study compared to people who received the placebo. In general, there was about a 6.5 month longer median survival in the group that was originally randomized to AMX0035, and the risk of death was about 44% lower in this group. This is all very exciting for the field of ALS and clinical drug development. As we bring more and more drugs to clinical trials, it's also important to think about newer clinical trial readouts and biomarker developments, especially in the earlier phases of drug development. If there's a way we can develop biomarkers that can tell us the mechanism of the disease that is driving the disease in different patients, and if we can tell if a drug that is targeting the disease mechanism is actually reaching the target or not, I think that would add a lot of value in accelerating drug development for ALS. One such biomarker is the PET target called as PBR28. You saw an earlier image that Dr. Sajid Vakili showed about inflammation in a patient with ALS compared to healthy controls, and there was a PET marker that showed increased inflammation in the brain compared to healthy controls. And that PET marker is PBR28. And our group has been using this PET dye for the last decade, and we've scanned over 200 people with this PET dye, and we have scanned ALS patients, many different types of motor neuron diseases, and patients who are at different stages of ALS illnesses as well. What we have learned is that when we look at the ALS group and compare it with people who don't have ALS, there is increased inflammation in the brain. And the inflammation is not everywhere, it's focal, and it is very robust in the region what we call as primary motor cortices. This is where the upper motor neurons reside, and this is a site of disease biology in ALS. What we also learned is that this inflammatory signal is more robust in people who have faster disease progression, and also in people who have more upper motor neuron signs clinically that manifest either as brisk reflexes when your doctor examines you using reflex hammer, or people who have more spasticity or stiffness in their arms and legs. Recently, taking this experiment, a series of experiments a little bit further to see if we can actually design a multi-site study where not every site may be producing the same PET dye, the PBR28 dye, not every site may have the same scanner as we do. Is it possible, is it feasible for us to combine the data across different sites as long as the dye is still targeting the same primary target protein, which is the TSPO protein, which is present within the mitochondria of inflamed cells within the central nervous system. We collaborated with a group in Belgium who were doing similar experiments using another tracer called DPA714. And we combined the two data sets and we looked at whether this increases the power of detecting inflammation in the combined data set. And this is the image that you're seeing that was published last year that shows that in comparison to each individual data set, the combination, when we pulled everything together using a state-of-the-art analytical pipelines that are automated and a standard template space to analyze these data, the power to show inflammation within the motor cortex and the supplemental motor area increases a lot. This is very exciting. What this means for the field is that if we were to develop clinical trials using imaging biomarkers, people don't have to travel all the way to MGH just to do these studies, but they could potentially participate in sites closer to their home if they have the capabilities to, if a site has a capability to produce and run these studies closer to home. More recently, PBR28 PET was also used in a clinical trial in Parkinson's disease using a drug called verdiperstat, which you heard about from Dr. Berry earlier. It's also in the platform trial. It's a potent myeloperoxidase inhibitor as an anti-inflammatory medication. What we learned in the Parkinson's disease trial is that PBR28 PET signal reduced by about 60% as early as 8 weeks. To inform that the drug is able to reach the target sites as early as 8 weeks, and this reduction was mostly seen in areas that are clinically relevant for Parkinson's disease. This is also a very exciting new development in the field of PET biomarker development, and using this information, as well as all the experiments that are ongoing in ALS, PBR28 is currently being used as a clinical trial readout in several early phase clinical trials. Currently, we have an exciting trial that's ongoing that using a drug called BLZ945, which is also a very potent anti-inflammatory medication. And this trial is sponsored by Novartis. And if anyone's interested in participating in this study, please feel free to reach out to me. A biomarker development program such as this requires a lot of people to collaborate and help with the development of this program. So it really takes a village to develop biomarkers. And in addition to colleagues at the Martina Center as well as at MGH, there are several patients, families, and funding supporters who have helped to continue this program currently. I'm going to shift gears just a little bit to think about, as we're developing biomarkers and drugs, we do need to expedite the diagnostic timeline for ALS patients. There is a tool that we are developing called as a Think ALS tool, which is targeting general neurologists or family physicians to create more awareness and educational resources for suspecting the ALS, as well as referring patients to multidisciplinary ALS clinics expeditiously. Currently, in the US, it takes about 12 months for an ALS patient to have a confirmed diagnosis of ALS from the time of symptom onset. This is a really long time. And our goal is to really shorten this timeline so that patients can have early access to FDA-approved medications, as well as early access to experimental therapies. And one of the barriers to early diagnosis is awareness at the first level of physician contact that patients may have in terms of suspecting ALS at the very beginning. Initially, it may be thought to be something unrelated to ALS, maybe carpal tunnel syndrome, or maybe a neck problem. And how can we improve this awareness and education among general clinicians to diagnose or think about ALS? And this is a tool that was put together as a national initiative that was led by ALS Association. And there's a working group that included many patient representatives, physician scientists, as well as industry partners. And we developed this tool that has information about when to suspect, when not to suspect ALS. And in addition, also provides resources in terms of when you do suspect ALS, how do we make the referral to a multidisciplinary ALS clinic expeditiously and really shorten that timeline to that first ALS consultation, and also provides resources for general neurologists or clinicians to find the nearest multidisciplinary ALS center close to where the patient lives. So that's my last slide. And with that, I will stop sharing. Thank you.
Wonderful. Thank you both for those presentations. This is when we're going to transition to the question and answer session. Really exciting things. I mean, I hope that everyone in the audience sort of gets the flavor that there are just incredibly exciting things happening in ALS, and we'll be able to dive into some of these a little bit more in just a minute too.
Okay, our first question of the day is, can you talk about some of the barriers to diagnosing ALS?
Thanks. So Suma, this really, I think, dovetails really well with kind of the last topic that you covered in your presentation. And I wonder if you wanted to start the conversation to talk about this.
Absolutely. That's a great question. And it is a hot topic. And I would say that first and foremost, the biggest barrier to diagnosing ALS is that we don't have a definitive diagnostic test that we could run to say this is ALS. For example, if you have to diagnose somebody with heart risk, you can run cholesterol levels. Somebody to diagnose with diabetes, you can look at blood sugar levels or hemoglobin A1C. We don't have a hemoglobin A1C test for ALS. And it still primarily is a clinical diagnosis, and there's so much heterogeneity in the disease manifestation itself. So, and that leads to delays in reaching the confirmatory diagnosis for ALS. And then to add to that, I would say, in addition, is that the biomarker development, which is not just a diagnostic tool, we don't have specific or sensitive biomarkers yet that would tell us, is this ALS? Could this differentiate from other diseases? And what type of ALS this is that a patient might be experiencing?
Yeah, I guess I would add to that you know, ALS is an uncommon disease, and so you know, there are estimates that primary care physicians see on average one case of ALS throughout their career. And of course, you know, some will see more and some may not see it at all. On the other hand, people coming into a primary care physician to say, you know, my arm feels weak, or there's something, you know, my voice has changed a bit, is much more common. And so this is, I think, why we really, just to underline your point, Suma, that we need sensitive and very specific markers that, you know, hopefully could be done easily and cheaply, so that we could really kind of find those potential cases early, but without raising the specter of ALS, you know, unnecessarily for people that are dealing with a different problem. And I think that's the kind of biomarker that we're looking for. And Gaz, I guess I would turn to you for just a moment and say, you know, we've worked on biomarkers together, and tell us a little bit about, you know, the work of looking for biomarkers and what that means in the lab.
Sure, yes. I mean, that's one of the things that we try to do in the lab in collaboration with our clinical colleagues, to use biofluids from patients, whether it's plasma or cerebral spinal fluid. You can actually use urine as well. When you know, we always try to go back and start with the postmortem samples from the patients. When we find a pathway that's dysregulated, like some of the ones that I was showing you today, and I'll just focus on tau for a minute. Tau is being used as a biomarker for Alzheimer's disease, or at least it's being developed as a biomarker for Alzheimer's disease. So one of the things that we did when we found that tau is also dysregulated in ALS is to measure tau levels in CSF or cerebral spinal fluid from ALS patients. So by doing those types of studies, but actually you need to have large sample sizes to make sure that this is happening across all cases, that you have the power, and that this is going to be something that holds up. Um, that's the kind of studies that we do in the lab. So once we think that something might be a good marker for disease, then we can go after it and measure it in different types of biofluids. Having said that, do we want something that's specific for ALS? So, as I said, tau is altered in a number of neurodegenerative diseases, including ALS. Does that make it a bad biomarker because you can measure it? It's altered in many different cases. It's probably not the best diagnostic marker, but perhaps we will have several different things that we can measure at the same time. If you drew blood from a patient or if you took their cerebral spinal fluid, if we had 2 or 3 different proteins that we could measure that would be altered, then that could give you an ALS signature type of biomarker. So those are some of the things that we're doing and thinking about currently. And I'll stop there.
Yeah, no, that's great. I mean, that's perfect. I think just understanding a little bit about what goes into developing a biomarker or thinking about a diagnostic biomarker is really critical. One place that this starts is having what we call biofluids. It's an unusual word for people who aren't in the field. It's kind of a term of art. And but basically taking blood and then we separate that into its fractions. Plasma is the liquid part. We all know about cells, but there are red blood cells and white blood cells. Each of those parts could be really important to us as we develop these. You mentioned urine. There are actually some potential biomarkers in urine that are being explored. But I think the key is that having these allows us to explore them, and so that our conversations about these projects often begin with what samples do we have. And so one way that people with ALS and people who don't have ALS can get involved is by donating biofluids or being involved in studies. And these are also studies that are often not particularly, at their outset anyway, often not the ones that sound the most exciting and are the easiest to fund. And so sometimes a little bit of philanthropy funding in the beginning can get us going into bigger projects that then are kind of more exciting to funding agencies. So lots of ways that people can be involved.
Okay, next question. Is ALS classified as an autoimmune disorder?
Yeah, I think I may start with this and then sort of hand off. I think, probably Suma does a lot of work in imaging the immune changes, and Gaz does a lot of work with looking at the biochemistry. Let me give a few thoughts on that just from a clinical perspective. Not so many years ago, 99% of people in the field would have said that the immune system was unaffected in ALS, and that it was not an autoimmune disease because the immune system was unaffected. I think that has really changed dramatically over the last decade, so that now the prevailing thought is that absolutely the immune system is affected. It's what we call dysregulated. So the wheels have come off in some way. It's not functioning the way that it should. I think it's important to say that there are very specific ways in which that is true, and in some, compared to some diseases, subtle ways in which that is true. It did take us a very long time, and we needed to have the right measurement tools to be able to see those abnormalities in the immune system. There are differences between ALS and other diseases that we would call autoimmune diseases, like rheumatoid arthritis or lupus or multiple sclerosis or Guillain-Barré syndrome. In that we don't know for sure in ALS that immune dysregulation is the match that starts the fire. It may well be like having gasoline in the house. If a fire starts, that's a very bad thing. And less like the match that starts the fire. And so that may be a fundamental difference between ALS and what we think of as classically autoimmune diseases. But those lines are blurring a little bit, and our language may need to adapt. Why don't we hand it over maybe to Suma to talk a little bit about, you can talk a little bit about your imaging and what that means, and then Gaz, you can maybe tell us about the molecular perspective.
I completely agree with you, Dr. Berry, about classifying ALS as an autoimmune versus a dysimmune process, and going back to what Ghazli, you had said earlier, that mechanisms are linked and they can overlap. No matter what triggers the disease up front, eventually there's a cascade of multiple different mechanisms that flow, and then there could be the immune system that is involved in many different ways in the disease progression or even perpetuating the disease. From an imaging standpoint, we're targeting inflammation, and the inflammation is of two kinds. One that pertains to glial cells that live within the central nervous system, but there could also be inflammatory cells or immune cells that flow into the brain from the periphery, from the blood, that could also be a part of that inflammatory process. The inflammatory signal that we are seeing on PET imaging and that correlates in many ways the clinical disease progression, as well as the clinical signs of upper motor neuron burden, again points to the fact that there is immune mechanisms that are involved, whether or not they are the key drivers of disease, clinical disease in an individual or not, it remains unsettled at this point in time.
And I would just add that going back to more of the peripheral biomarkers, such as cytokines or chemokines, things that are known to exacerbate immune response, it's kind of hard to know because there's so many studies that have been done looking at these markers in blood and cerebrospinal fluid, and we don't really have a clear answer on who are exactly the players that are, for example, increased that could cause inflammation. We don't really know that. And I think the immune system is really hard to hone in on, in the sense that many things can cause changes in our immune response, right? So you can have an infection, or even the food that we eat could alter our immune response. So as James said, we know that it's dysregulated, but whether or not it's the same type of autoimmune disease that we see with multiple sclerosis, for example, we're still not sure, because the key markers that would be elevated in autoimmune disease are not necessarily altered in ALS. But as James said, maybe we need to start thinking about all of this in a different way. And maybe there might be some immune component to that autoimmune components that we don't really know yet, but it's not your typical autoimmune components that you're seeing with your autoimmune diseases. And the biochemical molecular studies would suggest that as well. But I think we're just learning more and more about neuroinflammation and what's happening in the periphery versus what's happening centrally in the nervous system. So I think we'll probably have a better understanding of all this over the next few years, because people are really working on it now.
Question 3. Are there any natural remedies for ALS?
So well, I think we can all sort of chime in on this. I think this is a tricky one. First of all, I guess it depends a little on what we mean by natural, and also by what we mean by remedies. I think that there aren't, we don't know of cures for ALS that come out of naturopathic or sort of herbal medicine. However, there are many things that are not just medicine, not just sort of Western medicine, that are helpful for people with ALS. To start with very simple things, diet and weight, we think, are really important. It's very important that people maintain their weight, we think. And there are some studies showing that maintaining weight is a really important thing. Interestingly, we don't have a lot of studies guiding us to a kind of diet that's important, rather that people not lose weight seems to be important. There are some studies going on of supplements, everything from curcumin to TUDCA to alpha lipoic acid, to a number of acetyl L-carnitine, a number of other kind of supplements that are giving us some hints, at least early in studies, that maybe there's some benefit there. I want to be clear that there isn't proven effectiveness of these, but there may be, there are these trials that are showing us maybe hints. And so I think that in part the answer is maybe there are things outside of designed drugs that are from pharmaceutical companies that could be beneficial. I'll pause there and hand it off, and maybe we might have to go around a couple of times because this is a big topic.
James, I would add to what you said is, yes, this is a big topic, and the boundaries are a little bit blurred in terms of defining natural remedies as well, and to what extent, what combination, and for how long are all questions that come up very frequently in clinical practice as well. And the approach that I've taken also in my clinic is, as you said earlier, good balanced diet, healthy diet, maintaining good nutrition and weight are all important, whether that means in addition to taking the caloric intake of and proteins and carbs, should we also think about supplements, vitamins as well. And in general, the approach I've also taken is if there's no downside to some of the vitamins and supplements, there's no safety concerns with some of these supplements, and people with or without ALS are taking these supplements routinely, then it should be okay to take some of these, whether it's just curcumin or vitamins or NAD or other supplements. And then the role of microbiome and diet also is important, and that is being discussed a lot in neurodegenerative conditions, not specifically just in ALS alone. And that goes to, again, the type of diet, and again, eating a healthy, balanced diet that would affect the microbiome of an individual probably plays a role in regulating some of those immune processes as well, and helping to some extent. Gasly, I'd like to hear what your thoughts are.
I will just say a few things because I talked about reactive oxygen species and oxidative stress. I mean, I think we all might take a lot of things that already address some of that. So different vitamins like vitamin C, D, E, those are all known to be antioxidants. They could decrease your reactive oxygen species or coenzyme Q10. Inosine, these are things that you hear about, or maybe you can go to Whole Foods and like pick them right off the shelf. As long as James and Suma were just saying, as long as they're not going to be harmful, perhaps they could be, we know that they molecularly, they're hitting the right targets. There aren't too many studies on them. However, what we know about the mechanism of action of some of these vitamins would suggest that they would be good antioxidants. Or, for example, coconut oil can help with energy metabolism, for example, and we know mitochondria are dysfunctional. So based on some of the molecular pathways that we know to be disrupted and what these supplements do, then you could make an argument of how they could be more helpful, perhaps. But then again, keep it in mind that you should always talk to your neurologist, your clinicians before you take any of these things.
Yeah, I think that, so. I wanted to make sure that we got that in. That there are lots and lots of choices here. Some of these things that are even available over the counter have side effects, and it's really important that if people are going to go on a journey of looking for some of these things, that they do it in conjunction with a physician, and it ought to be I think a relationship where you should be able to talk about these things with your care team. So, really, 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 we encourage you to talk with your treating physician and treating team before making any of these decisions. Suma, I just wanted to touch on the microbiome because I think it's a really, really, it's a really sort of exciting developing area. Our close colleague Katie Nicholson is leading a project in this, recently published on this, that project has guided us along with others that have been done with in labs around the world, really guiding us to think that maybe there are some alterations that are important in the microbiome. And the project that Katie led really was started with philanthropic money and would not have come to the answers that it did without support from philanthropy. And it's a perfect example of a project that's impacting our thinking and wouldn't be here without philanthropic support.
Next question. Are there any signs or symptoms that indicate rapid progression or nearing end of life?
Suma, do you want to start us out here?
I can take lead on this question. I would start off by saying that AL is a very heterogeneous disease. Some people have slowly progressing disease. Some people have fast progressing disease. And almost everybody has periods along their ALS journey where there are long plateaus where the disease doesn't progress, and there may be some times when the disease may seem like it's rapidly progressing. And that makes it very difficult to have one measure or outcome that would tell us how do you define rapid and how do you define slow. Sometimes we think about in genetic forms of ALS, if there are known or variants or mutations that are known to have rapid course with multiple family members or multiple individuals across families, that may be associated with rapid progression. So that would guide us to say in terms of prognosis and what to expect down the road. But other times we may use some of the outcomes that we typically use in research studies, again, mostly as a research outcome measure, where we've created arbitrary cutoffs to say with ALS FRSR scores, if it is greater than a certain number, if somebody is declining greater than a certain number of points per month, that might indicate rapid progression. But I've also seen if you use ALS FRSR for disease progression, there may be some several months where there may not be any change in the score. And then there may be over the next 2 or 3 months, multiple points that somebody might have declined over a period of time. So it makes it very hard to put a single estimate, single measure that way. James, would you like to add?
Yeah, I think so. I mean, I think those are all really good points. I think one of the key points is that unlike the stock market, past performance does actually predict future performance a bit here. So that, when we get a sense of how things are progressing, that tends to be how things progress within an individual. There are these plateaus and stair steps, but we can take a step back and say, how steep is a staircase? And that, and I think, that can be an important point, but that only becomes obvious over time, is the progression kind of more rapid or not. I think one thing that sometimes we run into people fearing is that they will, that one day things will change dramatically for them, and that will be sort of the day when everything is different. That tends not to be the way that ALS progresses. Now, there could be other health problems that happen, and that can be unpredictable. But that's not the way that ALS tends to progress. That people are suddenly wake up and are unable to perform some function that they could perform the day before. So I think that's an important point as well. And that the tools that we have to help people with ALS actually do make a difference in people's lives, and though the sum of those tools is that people can have a better quality of life and live longer now than they did in the past. I think that's a really important thing to keep in mind as well.
Just two more questions. Can you please talk about resources that are available for patients with ALS?
Yeah, this is a big one, but also a really important one. Let's start out by talking, I think, about the key resource, which is the Multidisciplinary ALS Clinic. Now, not everyone is in a geographic location where they're able to access a multidisciplinary ALS clinic. Increasingly, at least over the last year, telemedicine has been a wonderful way for people to connect with multidisciplinary clinics when they couldn't before. And our hope will be that that resource will remain in place. I think, if anyone had an eye toward advocacy, some of the restrictions on telemedicine that were lifted for the pandemic may go back into place unless we really put some concerted effort into changing regulations around this. That is, we cannot practice across state lines, even using telemedicine, because our licensure is state by state. And without some big fix to that, we may be in a bit of a bind. I say this only because the Multidisciplinary ALS Clinic is really the standard of care for taking care of people with ALS. And it is the resource that brings together physical therapists, respiratory therapists, social workers, physicians, PM&R, neurologists, pulmonologists, orthopedists, psychologists, really a host of resources, and can really guide a person with ALS and their caregivers to kind of what to do in real time. And I just don't, we don't want people to have to go without that resource, and too many people do. It doesn't mean that, I think a lot of times people visit a multidisciplinary clinic periodically, and then their local healthcare provider carries out a lot of those plans, but I just think it's a really important resource. And I would never want to underestimate that. Having said that, there are lots of other resources, and Suma, I might hand it over to you to talk about some of the other resources that are available for people with ALS.
Absolutely, James. I completely agree about the multidisciplinary. I always say that that's at the converging point of multiple different resources that a patient can get access to or learn about. And even taking our own multidisciplinary clinic, for example, if somebody comes from a certain zip code and we're trying to figure out who might be the nearest physical therapist that we could refer them to, our physical therapists have built a network of PTs across various different zip codes in Massachusetts and around, and it would be very easy to guide, and also for them to be as a point person to guide the local physical therapist who may not know a lot about ALS, to say what is appropriate and what isn't appropriate in terms of goals and exercise regimens and so on and so forth. If I take a step back and also think about resources, it could be in many different shapes and forms. Sometimes resources can be in the form of social network resources for patients, in terms of support groups, in terms of local not-for-profit ALS associations or community resources that way. Resources can also be in the form of thinking about loaner closets to gain access for certain equipments and things like that, or even groups like IMALS or Gleason Foundation and so on and so forth, who help with a variety of different types of resources for patients. Also, resources can be in the form of online resources such as clinicaltrials.gov, where if somebody's trying to look for what type of clinical trial is appropriate for me, where I live or close to where I live, or for the type of ALS that I have. Or even a a website such as even the Healy Center website that provides so many different educational materials for, should I be thinking about a G-tube and what goes into making a decision about G-tube, or even a resource such as the MND Association, the NEALS. And so there are so many web-based resources as well that patients and families can take advantage of. And the list just keeps growing in terms of what might be all the types of resources that patients could have access to.
Guys, you use Twitter a lot for science, and it's a really powerful tool to share scientific ideas amongst researchers. I use Twitter for the weather, but I have a healthy uncertainty, I guess, about social media. I wonder whether you would talk a little bit about how you, as a scientist, and I think it applies to kind of how we think about this broadly for patients. How do you approach social media and science?
Very carefully, I would say. Honestly, I would say that I'm on science Twitter to really learn about like the latest findings. As you know, there's a lot more preprints coming out on bioRxiv and medRxiv, and I use Twitter as a source to be able to, really read this new science like hot off the press. It sometimes it could take a year before scientific findings could be accepted in a journal. That can then be consumed by all. So, being on there allows me to see that. But also, I use it to talk about some of the work that we're doing in the lab. I would say with everything social media, you should take it with a grain of salt. But I follow, I believe all the ALS foundations that are on Twitter. And, I think that they're a great resource. I know Healy Center puts out a lot through our MGH Neurology Twitter Twitter site or handle. So it's just really great to stay on top of all the events that are happening, all the different symposia, all the different types of educational programs that they have. Niels also has a great Twitter presence. So I really use it to find out what's happening, who's doing what, and what's the latest to find out.
Yeah. So it sounds like, yeah, no, I mean, I think it's really a growing resource, and kind of leaving us all to sort through how do we make all this work. But it sounds like you have trusted resources within social media that you kind of go to, and so
absolutely. I think all the foundations do such a wonderful job, and yeah, definitely to be trusted.
Yeah, I think that's sort of been our approach to thinking about this as well.
One other resource that I would point to, again, going back to the multidisciplinary clinic, is many multidisciplinary clinics, just like ours, they provide patients with welcome packets, and that may have a lot of information and resources all compiled in one place as well. Also not to forget the MGH ALS link that patients could subscribe to, and we constantly update our patients and families about new developments and new information as we learn in ALS as well.
We called it the link. We thought a lot about that name, and we called it the link because we really what we don't want is sort of isolation. What we really want people to, one of the big social determinants of health is connectedness, and so I think looking for those ways to be connected is really, really important.
And the final question: can ALS be prevented?
Also a really, really big one. The answer right now is that we probably don't have the tools right at this minute to prevent ALS, but it's we wanted to dig into this last because it is such an important area, and we are learning again through projects like the Dominantly Inherited ALS project, which is run by Tim Miller at WashU and by Katie Nicholson at Mass General, but other similar projects are enrolling people who are potentially at risk of a genetic form of ALS before they develop any symptoms, and doing that in a very, very careful and thoughtful way, partnering with people who are in the studies to really figure out how to do this well, so that we can look at, what may be the earliest horizon of change, and then know when we might intervene. Now, Suma, you talked about some remarkable studies that are addressing genetic forms of ALS. We have an increasing number of these ASO trials, antisense oligonucleotide. This is a new chemistry. Well, it's not that new, but new to be in the clinic and in neurodegeneration, that can target abnormal genes. And so I think it's safe to say that we are very hopeful that in these forms of genetic ALS that we may be able to prevent the disease before it ever starts. Gaz, you talked about Stathman-2 as a linchpin sort of molecule that brings together lots and lots of the different pathologies and things that we think are going wrong. And it is a genetic target, so that even in sporadic ALS, where we don't think there's a genetic mutation, a single genetic mutation causing the disease, we might be able to use those same tools to at least treat it very, very early. Preventing it, though, means that we have to predict it, and that's still a challenge for sporadic disease. So
I'll pick up where you left off and say, yes, today we cannot, but I think, projects like Project Mine, like the big genome studies, or the ALS Knowledge Portal, now Answer ALS, like there's so many big groups really doing a lot of sequencing and trying to find these mutations that could be causing disease. Even if we don't find like another big gene like SOD1 or C9 or 72, which mutations in these genes would cause ALS, it doesn't mean that there aren't a number of genes or a genetic loci that are mutated that could be linked to sporadic cases as well. We just haven't uncovered those. So I think these large genetic studies are really helpful, and people are still looking at them and mining them and trying to find these needles in a haystack. I also think that there is an environmental component. So it could be a gene X environment effect that's happening. So we know of, toxins that could cause ALS or ALS-like symptoms. We know perhaps, a connection with traumatic brain injury. So there are some things in the environment that could also be causing some of this. We're just starting to understand some of it. So today we cannot, do anything about it, but with the studies such as DIALS that James was just mentioning, digging into the genetics a bit more, figuring out whether there's a genetic plus environment component to some of the sporadic cases, I think we can get there. I think we have better tools today than we did 10 years ago. Bioinformatics is a big deal now. Just really thinking about the genetics in that sense has really led the way. So, we only knew about SOD1 20 years ago, and now we have over 30 genes that are linked to causing ALS. So at some point we will be able to.
Because I would just add to what you just said that, the genetic testing, genetic knowledge is so important. And the first step towards prevention is really awareness of what genes are causing ALS. And that number of the ratio between sporadic versus genetic forms of ALS is continually changing, even over the last 2 decades. And there could be other genes within that bucket, is what we call as sporadic today, and that may be genetic in nature. And genetic testing is expanding, but I feel like there is an opportunity for advocacy as well as for philanthropy to actually expand the work in terms of genetic testing, or even getting the insurance coverage for some of these genetic testing. as well. Just within the past month, I can tell you in my clinic, we have had 2 patients with no family history have a positive genetic mutation, which was earlier called as a variant of unknown significance. But in learning that there are scattered cases who have reported the same mutation, we were able to actually call the lab and upgrade it to a pathogenic association. So the power really lies in numbers, and the more we test, the more we'll find out. But there is some hesitation in the community as well to get the genetic testing done because it's not covered by insurance for the most part, and that puts a bottleneck in terms of learning more as well.
Absolutely.
Okay, thank you everyone for that. Just before Dr. Berry, I ask you to wrap up, I'd just like to encourage everyone to visit our virtual event booths where you can directly interact with biopharma representatives via video chat, as well as learn more about the therapeutic offerings and clinical trials. Just go to the link below this video on our website. On behalf of the McCourt Foundation, thank you everybody for being involved on this session. Thanks to everybody today who have been involved in all 3 sessions. We've just gone past the 6-hour mark. So it's been a fun day. We've learned a lot. It's been fantastic. And I'll hand it over to you, Dr. Berry, just to wrap up. Thank you very much.
Well, knowing that we have 6 hours behind us, I'll be brief. This was really wonderful. We're so grateful to have the chance to sit down. It's fun for us to talk together about these really big issues. I mean, these are incredible questions. I hope we sort of were able to paint the picture that there is an incredible amount of progress in ALS. Really, really hopeful things happening, and happening faster than ever before. And that to make progress, we really need to partner. We need, we need people to be willing to partake in these research projects. Many of the most cutting edge science projects depend on philanthropy to get going. We're all really dedicated to curing ALS. So thank you for participating today, and thank you for everything that the McCourt Foundation has done to bring us together.